Well, welcome everyone to another exciting edition of the Magic Sandwich Show, and I'm joined today by two of the regulars, Thunderfoot and Concordance. I'm going to keep the announcements to a minimum at this stage, but we'll just remind you that it is a call-in show, and if you would like to appear, send a contact request to Magic Sandwich Show, all one word, include in that, please, the gist of the question or topic you would like to talk about. There is no specific topic that we have today. I suspect we're going to be talking, at least to start with, about science. And I know that you can't say too much about it, but Concordance, you had an exciting week, I believe.
Yeah, we had a small breakthrough in a collaboration we're doing with a medical center in Houston regarding being able to diagnose residual disease in cancer. It's a major discovery. Unfortunately, I can't talk in any detail about it because it's part of a non-disclosure agreement that I had to sign. Once that embargo is lifted, I'll be able to talk a little bit more about it in general terms, because I don't want to give away too much about my personal identity.
Well,
I'm a dino, right?
You don't give too much away in your avatar, but all right. Let's talk about a video that you put up this week concerning, I believe his name is Brad Harrub.
That's correct.
Can you tell those who may be watching and haven't seen your videos who this person is and how it came about that you're doing what will be a fairly lengthy series of videos about him?
Brad Harrub is someone who makes his living going around selling books, selling educational material. He does have a PhD, so it is Dr. Brad Harrub. He's been a dim bulb of the week, I think, or a creatard of the week with Wildwood Claire's channel. He has a PhD from the University of Tennessee in Memphis, which is the smaller medical campus there. And he has all the trappings of that PhD. He has a single scientific publication in a very low-impact journal that hasn't been cited only by the co-author and his advisor during his graduate program. He has all of the looks of someone who tried out science, managed to squeak out a PhD, and then just walked away from the whole thing. Because at that point, most people would be looking for a postdoctoral fellowship or some sort of a research posting where they would then be very productive in publishing papers. This guy obviously walked away from the whole thing and went into Christian publishing and creationist literature. So what I'm doing, I attended a talk of his back in August at the local Church of Christ, where, by the way, what's his name? Don McElroy, who was the Texas Board of Education chairman during the textbook adoption, the one who said, somebody's got to stand up to these experts. Just a complete idiot. And he was also there, so I had a chance to be in the same room with the guy. I always assumed I would just spontaneously explode. And just going through the arguments. And the reason why I'm doing this, and a lot of people have said, why are you wasting your time on someone of this low caliber? Because the arguments are the usual creationist young earth arguments. But I think that I want to go on record answering these things as a scientist, as someone who values education. I want to put it out there. Look, when someone says this, here's why it's wrong. I can't remember who said it, but somebody said any science you can't explain to a 7-year-old is probably bunk. And I think that's what I'm trying to do here, is basically explain why Harrub's bad logic is bad logic, where he's gone wrong, where he's missing the science.
I think the one that you posted this week was part 3. And what I found very disappointing about it, not about your video, but about the quality of his argument, is that you had to deal yet again with the old canard of you never see a dog produce a cat. I cannot believe that anyone who claims to be educated, has a PhD, can possibly run that argument. Do you think that he is being knowingly dishonest when he comes up with this crap?
I think that there are psychological mechanisms in place where he can't bring himself to not believe it. And I think that you can get a PhD without learning a gosh darn thing. And I think he's one of those people. I think that he has managed to put the right answers down on tests. He's managed to do a very minor research project and shown that he knows how to physically do the things in a lab that have to be done to do research without letting any of it sink in, without letting the critical thinking sink in. And I suspect that's why he was so unhappy and why he didn't pursue a career in, say, cancer research or disease outcomes or epidemiology, something that would have contributed a little more directly to human health and welfare. And instead, now it's all about him and his creationist beliefs and, padding his pocketbook. He sort of sold out his critical thinking, what little he may have had, in order to turn this into a career. And what he's doing is so much easier. It would be so much easier if I could leave all the stuff in the lab behind and just go around talking about what I think or making these silly arguments or just entertaining people, basically. It would be a lot easier job than what I have to do every day. What Thunder has to wake up in the morning and go do, and the 10, 12-hour days in the lab are not very profitable. I imagine him going to a 30 or 40 talks a year is a pretty nice career.
But what I find inherently dishonest, and this must be dishonest, is that he purports to be in a position to speak about evolution when he quite clearly doesn't understand it. I mean, anyone who could think that a dog could produce a cat clearly doesn't understand evolution, because if they did, they realize that would actually completely fuck up the theory of evolution. So I'm sorry, I think that he must be being somewhat dishonest. Anyway, we'll move on, because I think that he was introduced at the speech you attended as someone that had too numerous scientific papers to mention. Explain that to us.
Yes, he's of course including everything he's ever written for, his own Focus Press. He's formed his own little press organization where they print off, and it was all for sale, of course. He was invited to speak by the local Church of Christ, but there in the back were, sign up for our mailing lists, buy our CDs of this guy talking. Here are coloring books for kids. Here's a textbook that we wrote for homeschoolers. And we talked about homeschooling last time being a big creationist thing. If you want your kids taught proper biblical science, those two terms are so funny to put together. Taught the proper biblical science, you can buy it directly from Focus Press, which is just basically Brad Harrub. But I'm sorry, what was the question?
You questioned him about the fact that he claimed
he was introduced.
Publications. How many is too numerous to mention?
Yeah, apparently too numerous to mention is his one paper he wrote as a grad student. And then everything since then, of course, has been a creationist article on a blog somewhere, or written down on paper and then sold. So he's basically listing his coloring books, I suppose, but his scientific publications, number one, and that is they number one. There is one of them. When I challenged him on this at the end, I said, too numerous to mention. I plugged your name into the scientific database of all published literature, and it spit out one article. He said, well, there's another one. There's two. I said, well, it doesn't appear in the database anywhere. Well, I'll have to give you my email, and I can send it to you later. All right.
The idea that two is too numerous.
Too numerous.
Thunderfoot, I saw when Concordance said pretty much anyone can get a PhD. You were nodding. Is this an act of modesty?
What, that anyone can get a PhD?
Well, you've got one. I mean, are you being self-deprecating?
I wouldn't say that anyone can get a PhD, but it's certainly having a PhD doesn't make you a good scientist by any means. PhD is basically just showing that you can actually do independent research. After that, essentially, people judge you by a track record, which is typically how many good discoveries have you actually managed to pull together. And in that sense, one peer-reviewed publication makes you a poor scientist. I mean, there are people who go into things like communication. And obviously, if you're going into the communication side of more teaching science, then you wouldn't expect to have this great publication list. But in those cases, you certainly, it would be really very dishonest to suggest that this great researcher with too many publications mentioned. Interesting point, though. I mean, this guy, you think that he is compartmentalizing, that he does sort of believe things that he's saying. Do you think that's true with the Discovery Institute?
No.
Do you think?
I don't, and I say that
Can I just before you answer that question, can I just remind people, if you'd like to join us, send a contact request to Magic Sandwich Show. We have no one at the moment, so you will get pretty much straight on. Sorry, Concordance, do go on.
I didn't interview them. I did a Q&A with them at SMU. And I asked a very simple question because I didn't want to get – I wasn't there to be confrontational. But they obviously had missed gene duplication. And I made a video on it. You guys can all go watch it. But you can hear what's his name? Richard Sternberg. You can hear him start to give the correct answer, the scientific answer. And then he backpedals. He realizes what he's done. He's conceded the actual mechanism by which they are pretending doesn't exist, that there's no way to create new information.
Implicit information, yeah.
Yeah, and so I said, well, what about gene duplication? Well, gene duplication is very important. It can create new functions, and of course is the root of neofunction. I mean, but nevertheless, and he uses nevertheless, so that's my favorite moment of the whole thing. And then he starts backpedaling. No one has ever actually demonstrated this as far as he obviously knows the right answer, but it is his paycheck. So I think Brad Harrub honestly believes somewhere deep down that what he's doing is the right thing. So even if it wasn't what he was taught, he's brought himself to accept this stuff. I know for a fact, just from the look in their eyes when they saw Richard giving the right answer, and at that point, Doug Axe stepped in to save him and sort of interrupted Richard because obviously he was going down the wrong path. He was leading himself into a dead end. They obviously know that they're wrong, but it's their business. They're like the tobacco lobby talking about lung cancer. You know, that's what they're getting paid to do. What do you think, Thunder?
With the Discovery Institute, they certainly have that sort of professional sleaze feel about them, that they are selling something. Because again, in terms of research, these people don't even appear on the radar. But I mean, the other thing is they seem to have dropped off the radar. You know, so they had they sort of had their flash in the pan, probably in the early 2000s, when they put out all this media, which someday I want to go back to it and essentially just rub their noses in it, because they made all of these very bold proclamations about how evolution was on its last legs. It was a theory just about to collapse. And that intelligent design was the only real viable alternative to any of this. And now just to compare this, what it's like 10 years later on. And they've just got so much humble pie to eat.
These are the people that talk about information in DNA, aren't they? Amongst other things.
You know, it's funny. They are creationists. That's sort of their fundamental. But their scientists are the least creationist of all of them. You hear young Earth creationists saying, "Well, I know who the intelligent designer is," and they try to borrow that language and sort of clothe themselves in this slightly more sophisticated form of creationism. But if you listen to Michael Behe, he is not a creationist. At most, he is a theistic evolutionist.
He believes in all Earth, common heritage. As far as he's concerned, there are just little bits here and there that you need a designer for.
Exactly. And if it were Behe, and Behe were the only member of the Discovery Institute, I would be interested in hearing more of what he has to say, because he's talking about front-loading mutations and phenotypic plasticity. These are scientific concepts, and they're areas where evolution needs the theory needs to adapt to include some unusual phenomena. And in that case, they're sort of the loyal opposition. Michael Behe, I have very little problem with the guy as long as he's sticking to the empirical science. It's everyone else in the group. It's all those non-biologists that make up the Discovery Institute. And they take this little kernel of, well, here's an unusual phenomenon. Here's an anomaly. Let's turn that into a reason to believe why the supernatural is the only explanation for this.
Now, let me just bring that over to sort of somewhat related thing that was in the news today about GM foods, where they claimed there was some study where they'd fed rats GM food and regular food coated with pesticides, and the GM food gave them more tumors. Did you see that, Concordance?
I did. It's a French study. I'm sorry. Let me get the phone.
Okay, I'm curious to hear your take. I mean, I can give my take on it first if you want.
I think that's probably a good idea.
The thing is that no sensible scientific study has on its front piece pictures of rats with giant tumors, right? That just has scare tactics written all over it.
Yeah.
You do a little more scratching around and you find out that this was actually essentially done by a rival group. So it's a company that makes pesticides, and what do you know that using their pesticides to actually save the GM. The sample sizes they had were tiny. With it's something crazy, like only 10 rats in their sample, in a standard sample. Now, when you get down to that, one or two small number statistics can make very big changes to the outcome of the study. And all of those things, I mean, there are genuine conflicts of interest to be had with GM crops and abuse of market position and abuse of sort of science to mislead people and such like. But in this case, I would suggest that most of what was coming out of this, it was, as far as I could see, a fairly simple attempt at scare tactics that you show rats covered with giant tumors and it's associated with GM. That's all you need to know.
So there are some red flags too in how the study was conducted. The kind of data that they produced, I don't want to get too technical, but the standard deviations are not given properly. They're given for the group, but not for the subgroups, which looks a lot.
With sample sizes as small as they're dealing with, can you even do any sensible statistics?
Well, that's the problem. And the whole thing hinges on an analysis of variance, an ANOVA. And the way that ANOVA was conducted, it fell just on the right side, which whenever you have a small effect size and no good mechanistic data, no understanding of how this happens, there's a very great chance it's a false positive. Now, I am perfectly prepared for Roundup Ready soybeans to cause cancer. I am perfectly prepared for that outcome if it happens. But this study didn't answer the question. It certainly titillated a little bit. It certainly got the news. And I strongly suspect that was the ultimate goal of the study. But given the source, as you said, given the type of data analysis, we can draw.
It's not just that. They had the video of the cancerous rats. This thing had prepared for TV written all over it. As in, would you actually have in these studies video available of the rats? Why would you even need to see what a rat with cancer looks like? There's loads of examples of that. Why is that now suddenly relevant? I mean, presumably, and that's the other thing. Apparently, the rats that they used had a propensity for
memory tumors.
Memory tumors.
Yeah, especially when the
what is a memory tumor?
Boob cancer.
Tits. Boob cancer. There we go.
I thought you said memory.
Memory.
Yeah. Sorry.
Whenever they're fed,
I do know what memories are.
Whenever they're fed unrestricted, whenever they're given as much food as they want, they tend to get obese or fatter, and that increase in adipose tissue growth can sometimes sort of carry itself over into memory tumors, and that's one of the outcomes that they measured. But again, they sort of collapsed those into subgroups. I read the study. My company, and for the sake of full disclosure, has a pretty big vested interest in GMO testing. We don't do any GMO. We test lots of food, or we make the technology used for testing lots of food for the presence of a GMO at a rate of something like 1 in a million soybeans. We can detect that. So we were very excited. The company was, I think our stock went up pretty substantially on that paper. So somebody's making money no matter what happens. If they find GMOs are safe, that makes the Monsanto crowd very happy. If GMOs are unsafe, that makes companies like mine very happy in the diagnostic testing divisions. There's money. If you want to say, oh, follow the money. Dude, there's money on every side of every issue. There always is.
And you're sat in the middle with a smile on your face.
Yeah.
Thunder.
Just so we can maybe be clear on what the potential risks of GM are. Let me just say, I'm very much an interloper to this field. So when the idea of genetically modifying these things, you essentially insert genes that will essentially produce an internal insecticide for the plant or similar. So because it's actually creating different proteins, there is potential for it to have. So, for instance, if you were to put a gene in that produced strychnine or something, then obviously this would make it very toxic. However, just because you're inserting genes doesn't necessarily mean that it will become toxic. I mean, eating the DNA itself will have no effect whatsoever because it all gets digested anyway. So is that right that the only potential hazard that you have here is essentially from eating the extra proteins that have been created by the genes that you splice in there? Is that right?
I wish it were so simple, because that's what we thought 10 years ago. But the problem, of course, gets more complex. As the more we study it, it keeps going down. The complexity increases. The protein, yes, and the protein is not like strychnine. It's a protein taken from a bacilli. It's a Bacillus thuringiensis, I believe. It's BT, which is a pathogen of bugs. And it's a pathogen. It causes disease in the bugs, or it harms the bugs through this protein, which causes damage to insect cells, but not to mammalian cells. And it's a free-living bacteria that some crops actually have on them that will protect them. So in the old days, we would take this bacteria and spray it on the crops. And I put dunks out in my lake that contain this bacteria, this bacillus, to kill mosquito larvae. So it is sort of a natural product. We've simply taken that harmful substance to the insects and put it inside the cells. So you've eaten bacillus, you've eaten BT, probably in a lot of organic produce in Europe, has the same bacteria in its native state. It has that same gene and that same protein. When you plug it into the crop, now it's inside the cells. You can eat the DNA, and the DNA is not getting in you. I mean, obviously, if you ate a banana and you got banana DNA throughout your body, you're not going to turn yellow and have little black spots and bruise. And I mean, come on. But we do know that some of the regulatory microRNAs can be found, some of the little nucleic acid stuff from inside a plant, when you eat the plant, some of it does end up in your body. Fortunately, you have all these defenses against foreign DNA. Otherwise, we'd be getting viruses from everywhere. But that's always a possibility. So the companies that do this stuff, I know they want to make a profit, but they also want to avoid lawsuits. There is a vested interest in the case of Monsanto or whoever, and I've no problem saying that they're an evil corporation if that's the kind of requirement you want to make. But they have a vested interest in not getting the pants suit off of them for killing 6,000 people or 80,000 people, whatever it may be. They do try to make sure that their products are safe. People have been eating this protein and this DNA in its bacterial form for decades. And now we're just saying, well, okay, we're eating the same protein and the same DNA, but now it's inside the corn cell. That's the only difference. So that's why it's not biologically likely that these things are oncogenic, simply because we know they don't bind to our DNA and produce the kind of changes that lead to cancer. I love talking about science.
It's fun, isn't it?
Well, apparently, I don't know whether the audience doesn't enjoy it or what, but still we've got no one wanting to talk to us. If you would like to join us and talk about these issues that we've been discussing or any other, please send a Skype contact request to Magic Sandwich Show. We will get you on. This quite often happens in the first half of the show. No one wants to talk to us, and then everyone suddenly in the second half starts sending contact requests. So get in early. Book early to avoid disappointment. Thunder.
Yeah, I mean, I just wanted to say that this is one of the things that, I watched a Young Turks piece on this earlier. And yeah, I really quite like the Young Turks, but their piece on the GM I thought was really missing in a lot of places. I mean, one of the ones they were sort of criticizing Monsanto for was saying that, when they're actually genetically modified this, it's essentially the same thing, which is true. But, those small differences, sometimes those small differences are really important, and sometimes they're not. I mean, for instance, whenever you actually get the stuff reproducing, you're going to get some mutants in there. And those are substantially the same. So, I mean, there's no real point in going crazy with the regulations there. So sometimes genetic modifications are absolutely benign, and there really isn't any need to go and do all the testing. However, sometimes it would be probably not that. The cascade effects in biological systems are always much harder to predict than you think. But I would expect that it's not that different to put in a gene for a potent toxin. In other words, you can put in something that is toxic to people just as easily as you could put in something that's toxic to insects.
Yes.
So sometimes very small differences matter, and sometimes they don't. And that was a point that I thought the Young Turks really missed.
Well, someone's posted in the comments on Blog TV that I think he expressed it as the elephant in the room on GM foods is the fact that we require it, given the ever-increasing world population and requirement for food. Would you both agree with that?
Can I say something about that, actually? Sure. There are two real modes. I'm not worried about the safety. What I'm worried about are the two uses of GMO. One is to ensure that first world farmers with access to the best technology have no problem competing against third world farmers who have no access to this technology. That is very much what Roundup Ready is. It's a technology to make the rich richer. I think if you're going to protest GMO, that's the aspect that needs to be focused on. The safety, I don't think there's a compelling argument to believe that just because you're not eating the bacteria, you're eating the bacterial protein in the crop is not a very compelling reason to believe that's less safe. But some GMO technologies could help level the playing field. Now, which of those two do you think is commercially viable and has a world market? Obviously, it's the one where the people with all the money can get even more money, can get higher crop yields, or can prevent competition from third world farmers. There are nonprofits who create GMO crop tools, if you will, to make non-arable land usable. So that food can be grown locally. The problem is not that we can't produce enough food. The problem is we can't afford to ship the food from Kansas to some place in Africa where it's needed the most because of the fuel costs, because of the petroleum required to move that food. If we could grow crops locally, it displaces the need for these imports, and that would help to reduce the poverty issue in Africa. They could become more self-sustaining, and that would lessen the economic burden that they put on the rest of the world. So that's something that's more about leveling the imbalance, whereas Roundup Ready and Monsanto's products are being sold to rich farmers to help maintain that hegemony, that power imbalance. So I see two very different uses for the same tool. In the same way you can talk about fire and whether it's used by an arsonist or whether it's used as a fire break to maintain the park system, or guns when they're used by police versus being used by criminals. If you want to protest Monsanto, go ahead. They've done some pretty unethical things. But don't confuse all uses of GMO into that hegemony maintaining technology use. It's got a lot of other uses. Yeah,
I've got, I think, 3 issues with this. The first is very similar to your point. It's more the business practices of people like Monsanto, which it has to be said are sometimes on pretty shaky ground. But not only that, a lot of the science that they put out, has that appearance of big tobacco. Now, whether that's just their PR department or what, I don't know. But it looks like it's just got conflict of interest written all over it. Monsanto doing all this research to show that GM is safe. It would, if they were making big profits off it, it would be easier to take them at their word. I don't have really any problem with eating GM foods in that all the stuff that goes in just gets digested anyway. And unless there's actually a problem with the plants actually producing some real toxin or something, I don't really see the potential for harm there. And given that, thousands of tons of this stuff's already being eaten, if there were significant health effects, we would know about them already. The third one is maybe my only real concern with GM, and that's the potential for genetic leakage. It's all about how genes flow around in the wide world. We know, presumably, that there is very little horizontal gene transfer. You can actually get these things can drift into bacteria, and bacteria can move around and that sort of thing.
Can you just help me with that, Thunder, and perhaps some people in the audience who are equally as dumb as I am? Horizontal gene transfer, what is it?
So, I might be wrong here. This really is my field, but the usual one is vertical gene transfer, which is mommy and daddy pass on their genes to their kids. The horizontal is where, so bacteria sort of do this. If you get a load of bacteria together, they can sort of exchange DNA fragments with each other without actually having children.
So it's equivalent if I exercised some frottage with you, there could be some transfer of genes material between the two of us, but on a bacterial level.
This is the thing, right? But with us, unless that genetic material actually gets into, this is going to go wrong, unless that genetic material gets into the bits that produce babies, then there's no point in it. Because if you actually get your DNA into me, then if I die, that stops there. Yeah.
You heard it here first, folks.
But say, for instance, with the retroviruses, so this is the way, if I get something like AIDS, where the HIV virus actually injects DNA into my genome, and some of that will actually happen in my gonads, and if that happens inefficiently and the virus doesn't become active in those cells, and they're then used for reproduction, then the DNA that was in the virus has now essentially become part of the genome of my children, right? And I think that's the only mechanism that I know of. I mean, do you get things like that with plants, concordance, or?
Oh, totally. Totally. Actually, you know, people have no idea. I imagine the average person just has no idea. We are made of viruses. We are hugely made of viruses. Viruses that have jumped from one species to another and carried that material with them. So we used to think of this sort of tree structure, and now the tree is so interlinked between all species because of viruses and plasmid transfers and all these sort of fusions of genomes. At least 10% of your genome is of recent viral origin. 10% of your genome is made from viruses very recently. And then once it gets into your genome, it gets shuffled around because some of those viral proteins are still functional. And they're sort of infecting you with more of you. And that creates all sorts of weird outcomes. But you are made of virus.
Somebody's asking concordance whether this is the same or makes up junk DNA.
Junk DNA is not a scientific term. Yes, some of it is considered non-functional or non-coding. But in some cases, it can have what's called a positional effect. This is just one of many ways it can function. It can hop down, this virus part, can hop into an old, quiet, fossilized part of your genome and reactivate it. And I say you as though you were evolving, but the entire population is what I'm talking about. This has happened hundreds of times in the primate lineage, where a virus has reactivated some latent gene, some old fossil of a gene that radically altered the way the organism looks or acts. There's so much going on. Your genome is not a library. It's like a factory. It's more like people reconfiguring things all the time. On an evolutionary scale, and that's where it gets a little confusing for most people because they can't. I keep jumping between terms where I'm talking about over the last million years versus someone walking down the street in the course of 30 minutes. Course of 30 minutes, your genome is fairly constant. But when we start talking about the human lineage or plants, potatoes, or strawberries, strawberries are a highly polyploid, meaning they've had their whole genome duplicated multiple times. Some of them are like 16 in. They're 16 copies of every gene out there. And that changes the way they behave. It changes their sweetness, and it changes how quickly they ripen. So all of those things, and some of that is human-induced, some of it's wild. Viruses also play a part in all of that. They hop around, they turn genes on, they turn genes off, they move genes out, they turn them into being linked to another gene. So there's a whole lot more out there that unless you want to go so deeply into this topic, that you'll just never understand.
Well,
and this would be a great segue into the new encode series of papers, which we can talk about after DPR.
Yes, well, I was saying that there seems to be a lot of questions being posted in the chat. I'm not seeing all of them, but just to remind people, rather than posting in the chat, you can come and ask Concordance and Thunderfoot in person by sending a contact request through Skype to Magic Sandwich Show. So your segue was somewhat interrupted, Concordance.
So there was a series of papers, and it was controversial, directly related to junk DNA. And this goes back to your question, Thunder. What it was is it was publicized as identifying finding a functional element for 80% of the entire genome. Now, the more traditional way of looking at the genome is that 1% of it codes for protein. Those are the genes. Then another 4 to maybe 8% could be considered semi-functional, regulatory. It changes the way the other the 1%, the most critical 1%, acts. Yep. Now they're saying, 80% of the genome does something. Had you guys heard that?
No, I mean, the epigenetics is, I think, a very big step forwards in that, so what you get with DNA is some of it translated into proteins, essentially. But it turns out that how the DNA is actually all folded up can also affect the expression of how much protein, how frequently that gene puts out a protein. And that can be as important as having the sequence there in the first place. And be, I mean, I can see how that might, get you from whatever 1 to 4%. How you get it up to 80%? I'm not sure. I mean, is that basically saying?
That's what really makes it sad. All right, because of course the intelligent design creationists are saying, "We told you so. We told you so." 80% are the percentage of nucleotides that show up in some assay of something somewhere. And even then, it's based on an assumption that they're missing 95% of all the functions. What they actually found was somewhere between 8 and 16% of the genome has some function, something that it does. But it could be something as simple, like you're saying here, as providing some structure. And then they assumed that they're missing 95% of it. They're only seeing 5%. Therefore, multiply by a factor decided ahead of time, we come up with an 80% number.
Yeah.
But that's not actually true.
Yeah. One of the things that I understood is if you've got a lot of functional genes, you need a lot of filler between them, because quite often when your cells are, mommy and daddy's DNA are coming together, they do this thing called crossover, where these sort of chromosomes all sort of break and reform. And if that happens in the middle of a gene, then that can be fatal. So by having more filler between the genes, you actually reduce the probability of that happening. So, I mean, just looked at like that, just having the filler there is actually filling a functional role. But what it is doesn't really make any difference.
Well, that's partly true. You mix two concepts there. The spacing between the genes isn't about homologous recombination. The crossing over you're talking about is never or is rarely fatal unless it's unbalanced. You don't get the same amount from mom and the same amount from dad. That can be a problem. But the bigger deal is the further apart these genes are, the more likely you are to get independent assortment. I'm trying to boil this down. You stack the beads on the string further and further apart so that when you go snipping with the scissors, you get fewer beads per snip. And that's kind of what it's about, is increasing the recombination distances between these genes so they don't co-inherit. They don't both come from mom and both come from dad. You can get a copy from mom and a copy from dad. They also soak up mutations. That's another really important one, is they soak up mutations. One of the real challenges to any argument like the one being made by the ENCODE data that everything's very functional is the fact that genome sizes vary so much. And I think it's T. Ryan Gregory, great guy, came up with the onion test. The onion has a ridiculous amount of junk DNA. I don't even remember the numbers. But let's say 8,000 times as much DNA per cell. I don't know if I'm even in the right ballpark there, but 8,000 times as much as a human cell. What is an onion doing that it needs that many more functional DNA bases than a human cell? It's an honest question. Anytime you propose some really high percentage of function for all this non-coding DNA, you have to explain why the onion needs so much and why the pufferfish, which has a much, much smaller genome, needs almost none. And then you get the amoeba, which has this incredibly large genome. Why does the amoeba need that?
Can I just ask you, when you say need, do you mean that it could not survive without it?
Yeah, it can't do whatever it needs done. You know, are the regulatory needs of an onion that much more than a human cell? Is there some explanation for why this variation exists in organisms that don't seem unusual, but have extremely unusual genome sizes? Why does an onion need to do its oniony thing to sit there in the ground and grow? Why does it need so much more DNA than a related plant?
Is oniony thing?
Yeah,
is that a scientific
onion
term?
thing. Yeah, totally. So the onion test has not been satisfied for this ENCODE discovery or this ENCODE paper. There is something else controversial about the ENCODE paper I think might be worth mentioning, and that is another data embargo. And we're seeing these more and more in research where people decide to publish together on these, what we call big science projects. Big science is where you get like $60 million worth of research at 18 research institutes. And everybody's working on a slightly different part of the problem. You know, these are the big projects. We're going to sequence the whole genome of 100,000 people. You can't do that in a single lab. So you have all these coordinating groups, but they have a data embargo agreement where none of them can publish until all of them are ready. And what that does is it puts, you know, some discoveries back by 2 years while we're waiting for one group in France to finish. You have that information not available to the rest of the scientific community simply because it makes great PR. It splashes the headlines when you have these really big papers. And the ENCODE results, I forget how many papers it was, all published simultaneously so that no one would get this, you know, sort of background information. This 80% number would come out along with all these papers all at the same time. The cost to science at the expense of making headlines, of public relations, and you know, making people feel good about, wow, they really did something, is something that a lot of us criticize. We're going to see it more and more, though, in research, where people hold off on publishing so that they can make the headlines above whatever Kim Kardashian is doing that week. What do you think?
I've just got a random. The last big science project that I saw was kind of funky. I've got to admit, I was impressed, not so much at the results, just at the technological achievements of what they'd managed to pull together. And they'd created an X-ray laser. It's a big facility. Now, I was stunned at that because I didn't even know that you could make an X-ray laser. And it's just some sort of great big long tube thing, and they sort of fire things down. That gets them their X-ray pulse, monochromatic, of course. But that was nothing. They wanted to look at the structure of, as it turns out, water. They had this jet that actually produced micron-sized beads. So you imagine a paintball gun on an automatic. Apart from these things are, a fraction of a millimeter in size and very regularly spaced, going out in this jet into a vacuum. And then these were getting hit by femtosecond X-ray laser pulses. And this thing was an absolute monument to science, triumphed only by how boring the results that they got from it were. But that was because they were looking at just water. I mean, the reason that they're actually building this machine is they want to do X-ray crystal structures on single protein molecules.
Awesome.
Right? So, yeah, because the reason you need the femtoseconds is because you're going to put so much energy through the thing that you're basically going to disintegrate it. So after this X-ray pulse goes through this micron-sized droplet, it's completely vaporized. But all that takes quite a lot of time. It turns out that bond vibrations are about femtosecond in time scale. So you can actually basically put as much energy into this as you want, as long as you get it short enough. So this is one of the big problems. Virtually everything that we know about crystals, proteins, we get from crystals, and crystallizing proteins is an absolute pain in the ass. So what they're going to try and do now is they're going to try and get proteins in these things. They blast them with the X-rays and you get your crystal structure, or you get your protein structure off of these things. But anyway, sorry.
I think people forget just how competitive too those projects are, because there's always some group somewhere who is maybe a month behind you. And so there's always this fear that if you don't get ahead of them, if you don't publish, then all the grant money will pass you by. No one funds the second project. No one's interested in the group that missed it by just a little bit. In that case, it's a bit like the space race. But now in the field of biology and chemistry and in all these other research areas. But at the same time, that rush, that constant pressure forwards sometimes creates bad science, and it creates cutting of corners. And that's why I think your professional scientists read some earth-shaking outcome, and they're always a little bit more skeptical and less enthusiastic, because you get burned so many times, and you understand the pressure these people are under. The gee whiz science, as I would call it, the stuff that makes the public newspapers, the newspapers of record, even if it's on page 7 of section F, always makes me suspicious, because it's so easy to get headlines from bad science. It's all mixed up together, and science journalism needs to clean up its act. I'm really tired of it.
Well, we'll have Dr. Lona Frank back on the show to defend scientific journalism. Seeing as this topic of conversation doesn't appear to be engaging callers, I'm going to move on. We've been on it for almost an hour, and it kind of leads on from what you were saying, Concordance.